Michele Barbeau, and I didn't recognize a lot of the names, but so I'm not sure if you've taken 3200 and 2200. So that's a neuroanatomy and the gross anatomy course. If you are in that course and I didn't recognize your name, I apologize. There are about 300 students in 2200 right now. So it's a lot of names to remember. Thanks for inviting me, first of all, to this. Haven't actually been asked really to share my research. Mainly my role in anatomy is as an instructor, but I do research as well. And my students are from the clinical anatomy program, which you may or may not know about. I can tell you a little bit more about that if you're interested. But basically, those students, it's a master's program. It's not a thesis program, but they do have a significant research project. So my research consists of these smaller research projects that are, some are connected and some are just sort of standalone. So what I wanted to do today was just to sort of share with you my kind of pathway, I've called it. So I did not start off university saying that I want to be an anatomy instructor. I actually had no idea that there were anatomy instructors. I hadn't given it any thought. I had originally started first year thinking maybe medicine, maybe I'm not sure what, but I definitely loved biology in high school. And so from there, I did my undergrad in cell biology here at Western. And so I was very fortunate to have a very small cohort of students. And we had several hands-on lab courses that we could take. And I learned a lot of skills, lots of things that didn't work, but you kind of got exposed to a lot of different techniques in that. And so I really enjoyed that. I ended up working a summer in one of the labs of one of the instructors and sort of working away at different things, trying to do tissue culture, things like that. So that was a great experience. When I finished my degree, I actually took a year off and worked for part of the year and traveled for part of the year. That's an entire other topic. It's a great thing to do if it wasn't a pandemic, but I don't know how feasible that is these days. So during that time, I kind of thought, what do I really want to do? And then I decided that I wanted to actually be a nutritionist. So I came back and did another undergrad degree, but I could do it in 2 years because I had most of the courses already. So I now had a degree in cell biology and a degree in nutrition. But then I decided I didn't want to be a dietitian, that I really liked working in the lab. So that led me to my master's in anatomy and cell biology. And I'll tell you about the work that I did there, but that was also where I met Dr. Pin, who's going to be coming after me and talking about his research. So it was a great time. So what I did there was the perfect combination of cell biology and nutrition. So I took care of pigs. So I had 32 miniature Yucatan swine that I fed this fatty diet for 8 months. And then I switched them to a diet that contained fish oil. So at the time, fish oil was this, it's going to cure everything, but it really hadn't been really rigorously studied, or the studies that they did have were sort of flawed. So they weren't really matching the oil. So it's a certain type of oil, and then they would match it with a fully saturated oil, for example. It's a monosaturated oil, and they were comparing it to palm oil, which is, it's not a good comparison. So the controls were not great. So we developed our own control oil. And then we looked at, so I had this background where I had taken biochemistry, I had taken some physiology, I had nutrition. So this was a perfect combination of all of them. So the research project involved biochemistry, looking at the lipoproteins of these animals. So I took blood samples from the animals every 2 months and measured the various, this is all the cholesterol, but I also looked at all the LDL, the VLDL, the HDL, all those things that are pretty common in your vocabulary these days. So we did a lot of different biochemical examination of them. And then we also, we had to unfortunately sacrifice these animals, and we looked at their aortas. So we looked at the accumulation of lipid in the walls of the aorta. So these images up here are, I had to dissect out the aortas of these pigs and figure out how to stain them. So I'm looking for a fat stain. So this is Sudan 5 that stains lipid in the wall of the vessel. So you can see all the red here is accumulated lipid. So this is before on the high fat diet. So it would be at the 8-month period. And then this is, I believe, after. When a lot of the lipid, although it doesn't look like that in this one, a lot of the lipid got taken away. So the lipid regressed from the walls of the vessels. The other thing that I did was I took little pieces of the aorta and looked at it under the microscope. So also combining histology and looking at the cells and what's happening here. So just for your interest, this is a section through the aorta. And you can see that it's all nice and regular, and there are smooth muscle cells here, and all of this white material that's all accumulated here are cholesterol crystals that accumulate in the atherosclerotic plaques. And you can see some of the cells have accumulated, especially down here, they accumulate lipid. So I was looking at using my histology background and sort of analyzing these. So what I had to do for the looking at the area of the vessels that were stained, I had to use different computer programs and basically trace around here to outline the stained areas. So I would get an area of the vessel that was covered by this this lesion, and then compare the high fat diet, or the when they were on the high fat diet, to when the regression period, to see if there was a difference in the area that was covered by that. So this project used a lot of the different skills that I had gained in my undergrad, and it sort of just brought them together nicely. Some of the other skills that you're not sort of directly taught, but that you certainly do gain a lot of experience in, is writing. So you are constantly required to write up your material. It's always encouraged for you to publish this material. So I was able to write it up in a manuscript, send it off, they send it back to you, you send it back. So all that sort of back and forth, doing the edits, which hopefully aren't too many. But the process of writing, I think, is very, very important skill for everyone to gain, that you certainly get a lot of experience with in a master's program, and certainly in a PhD program. The other skill that you get a lot of practice with is presentations. So you're constantly doing presentations throughout your work to your committee. So you have an advisory committee, you have to present your progress, and then they sort of help you figure out what's next, or say you're doing great, you should be doing, try this or try that. You also get experience presenting at both local and, if you're lucky, at international conferences. Again, that's not happening so much these days, but definitely there are virtual conferences that are going on it's a great experience. And certainly if this is the way we're going to be for a little while, you better get used to being in front of a camera. So there's a lot of sort of accessory skills that you gain doing some graduate work. So I did finish my master's, and then I went on, and I didn't think I wanted to do a PhD because I felt I wasn't cut out for it. Maybe I loved doing my master's. It was a great experience, but I didn't think I could be constantly writing grants and things like that. So I thought I would teach. So I got a Bachelor of Education, and then I actually returned and I kept doing research. So at this time I was also having kids. So I had little kids, so I was working part-time doing more research back with my master's supervisor, and I was an instructor in histology, so just part-time. But then an opportunity came up for me to coordinate the histology course, which I highly recommend, 3309. So I did that for a few years and really loved it. And then I thought, if I want to do this full time, I need to have a PhD. So after that, I enrolled in a PhD as I was quite a bit older than all of my peers, but it was great. So my PhD work was a completely different type of work because I had been doing this teaching in histology. We wanted to convert the histology course to an online course. We needed to expand our enrollment. We didn't have the physical space to do it, but there was a demand for this course, so we had to figure out a way. So we created an online version of the course, which is very similar to the model that we currently use for histology and our anatomy courses. So my PhD work mainly looked at studying how students use that course. So I went from working with animals to now working with students. And so there's a whole other set of skills that you have to get to do this type of research. One of which is there's significant ethics that you have to make sure that you're meeting. Your project has to be vetted through our ethics board, which other projects do as well. But when you're using students and you're an instructor, they have to ensure that the students aren't being coerced into participating and all these different things. So it's a good thing, but it's kind of adds a little bit of work. So let me just give you a little bit of a synopsis of what I did. So this is showing, I could see when students viewed the online lectures. So down here, we've got each lecture, and this is the attendance. So this is at 7 days after, this is 14 days after, and this is any time before the end of the course. So we could see that here and here, there were two different points where attendance really dropped off, the immediate attendance. And so that, interestingly, corresponded to midterm season. So this was right around the middle of the term. This is the sort of the halfway of the term, and here's term 2, midterm. This was a full year course. But we could see that eventually most people watched the lectures at some point. So these online lectures gave students the flexibility to view it when they wanted, but then most of them eventually watched the lectures. So you'll see here that not everybody did. It was never 100%. So that the next thing that we looked at was we looked at exam scores, and I took attendance in our face-to-face class and I took attendance in the online class. And then I looked at the time we had multiple choice exams with basically a few questions coming out of each lecture. So I could see which questions students got right. Did they attend the lecture? Yes or no? And so I was able to see that just by attending the lecture, and so you saw that we didn't quite have 100% attendance, but even if you read the material, so this would be you did not attend, just by attending the lecture, you had a significantly improved exam score. So just attending was beneficial. The online students had an opportunity to view the lecture multiple times, which some students did do. So I could see that they would maybe see it 2 or 3 times, or maybe just parts of it. However, that did not correspond to any further increase in how well they did on the exam questions. So it's just that first initial attending the lecture. And so that's one thing in my courses now. I really encourage students to attend the lecture, even if you can't really keep up with doing the pre-readings or reading afterwards. Just sitting there and basically hearing the material is still beneficial. So then I could also look at what sort of patterns of how students use them. And then I could also, so I'll just sort of highlight this because I could see how many students were watching for the first time the lecture 7 days before the exam. And so I could see that there were quite a few students who this I think was 7 days before the exam. So that's a bit skewed, but these are students who are leaving the lecture and then only watching it the week before the exam. So that also informed how I set up my courses. So now I have, you have to do a quiz every week just to sort of keep you on task. So all of this work basically informs how we set up our courses. So currently I have 3 students. 2 of them are doing a work with the occupational therapy program, and I have another student working with the physiotherapy program, mainly for physiotherapists. So they are doing what's mixed methods. It's known as mixed methods. So it's both qualitative and quantitative, gathering quantitative data. I also previously, not this year, but a couple years ago, had another student that was working with a medical device company. And so we did some clinical research. So I'll just sort of highlight a couple of things from those. So all of these were basically, we needed to solve a problem. The OT program, the students don't have to have anatomy to get into the program. And so we sort of, their marks are kind of all over the place. So they needed a little bit of help with that. And the physio, we wanted sort of to create a workshop for practicing physiotherapists because they don't get any pelvic floor anatomy in their program, but that's an area that they need a lot more work on, or it's very important for their practice now. So just briefly, our occupational therapy program, we sent a survey. We created a survey for practicing therapists, and we sent that out to them to gather what anatomical information do you expect your students to know. So we came up with some of the very important topics that were essential for their practice. From that, we created a knowledge test, and we got the students to do a pre-test based on those topics. And then we did a post-test after they finished the course to see if there was a change. So from this, we could see that there were there was quite a range in abilities, and that there were actually, we could get out 3 different sort of subgroups, and we were mainly interested in this low group, to sort of get them up to speed of all of the other of their peers. And then, so this idea of the low group is informing our next study on the OT neuroanatomy to help the students who struggle. How can we create learning modules for those students? And then, yeah, so this is just that topic. And then the medical device project was, we were approached by Bayless Medical because they had developed a device that was used for atrial fibrillation treatment, where they feed a catheter in and to reduce or prevent atrial fibrillation. So what they do is they feed a catheter into the heart. So atrial fibrillation occurs when the signals for the heart to contract don't work properly, and so you get the heart fibrillating, and this could lead to strokes. So what they do is they go in, they feed a catheter up, and they ablate. So they burn around the openings of the pulmonary veins. So when they do that, there can be some complications from that technique or from that surgery, where because they're using heat, it actually burns. You can see here the the esophagus is directly behind the heart where they are doing that ablation. So we used cadavers, and we inserted this medical device, and then we did CT scans, and my student took different measurements from the CT scans, created 3D models, and then he was quite computer adept. I could never have done this, but he created 3D models and used his computer skills to kind of create a heat map as to where the esophagus moved, to inform this company that we didn't want this device to actually make things worse by pushing the esophagus closer to the left atrium. So that is sort of my winding path through research. And basically what I kind of think is that research kind of gives you a set of skills, and that you're able to sort of combine all of your sort of basic knowledge that you've got, and then you also build on other skills such as presenting and writing, which are essentially very, very important no matter what you end up doing. So this certainly wasn't what I had planned from the beginning, but it's sort of where you end up, and it's all good. So just sort of taking advantage of opportunities that come along. So I'll stop sharing for now, and I assume that will sort of leave the questions probably to the end. I don't know how you want to do this.
Yeah, thank you so much, Dr. Barbeau. That was really cool. Thank you. Yeah, we were hoping that if we could leave questions to the very end, just so then you can ask for both you and Dr. Pin as well. Thank you.
Great. So am I good to go?
Yeah, you can go ahead whenever you're ready.
Good. So thank you as well for inviting me to give this talk. I'm going to take a little bit different slant than Dr. Barbeau did. Actually, it's great to see the stuff that she's doing, because I don't think I've actually seen, well, I definitely haven't seen that early work in a long time. And I wonder when the last time you presented it. It probably has been a while, but it was great to see, because I remember that, I don't know if we shared an office, but we were definitely beside each other with regard to the labs and the offices. So I'll give you a little bit of my story, just because there's a few points that I wanted to make. And I think that Dr. Barbeau has actually come up or said a few of the things, but I want to reiterate them. You know, the first thing is that where you start off is rarely where you end. You're always moving forward. And the reason I say this is has kind of been the track of my career. So uh, unlike Dr. Barbeau, I actually went right from fourth year into graduate studies. I didn't do a master's. I transferred to a PhD. So I was done my graduate work within 5 years. And then I was down in Indiana where I did a postdoctoral fellowship for 5 years. And then after that, I came back to London, and I started in 2000 as an assistant professor. And this year I'm going to be actually promoted to a full professor. So it's been a long, winding road, but it's been a nice one. The big thing that's been different is the work that I do. When I worked on skeletal muscle and try to understand why fast fibers were fast and why slow fibers were slow. And I went down to Purdue University to continue that work as a postdoc. And within 2 years, I was working in the pancreas, and I've never really looked back. So, you know, I've essentially changed a lot of the things that I do, but always going forward. And I think that that's the second message, is that you're always moving forward with whatever you're planning on doing. And so, as I said, I came back here. I started up a lab here. It has been not as diverse as Dr. Barbeau's research program. I'm mostly a cell biologist. I do a lot of histology. I do a lot of gene expression analysis. You can see from my title, I'm a part of the Children's Health Research Institute. I'm the head of the Genetics and Development Division there, and I'm an associate professor in all these different departments, which oddly enough doesn't include anatomy and cell biology. But what I want to point out here is that the work that's done in anatomy and cell biology, and the work that's done in physiology and pharmacology and in biochemistry, et cetera, is all very similar from lab to lab. And so you can work in all of these different labs and do very similar projects. And really the key is to find the person or the program that gets you most excited about the work. There are people who work, I work on pancreatic cancer. There are people who work on heart development. There are people who work on neural development and acquired brain injury and things like that. And you really need to find out who you want to work with and then kind of go from there. The other thing I want to point out, and you'll see some of this stuff, is that, you know, really what I do mostly now is I try to get people to collaborate. And when I say that, it's, you know, people in my lab with people outside my lab. And we do a lot of collaborative research in town, but you can see my backdrop. The backdrop there is in Marseille. I actually have an ongoing collaboration with the group in there and in CERN. I have a group collaboration with groups in Michigan. I have collaborations with groups in Toronto, and basically in a lot of different places. I got another one with one in Spain. So it's very collaborative, very international, and also importantly, translational. And so, you know, you saw from Dr. Barbeau's stuff that there was definitely this idea of like, how can we take educational research and translate it to the classroom? How can we take imaging and translate it to the way people are treated in the clinic? And so this is the type of stuff that we do as translation. So hopefully I'm going to talk or show you some of that. Okay. So now really about my work, or the work I did. And again, I'm not going to talk about specific projects. I'm going to talk about general themes. And so I work on pancreatic cancer. And if you know anything about pancreatic cancer, you know it's very deadly. This is the third most common cause of cancer-related deaths in North America, but it's like the 10th most diagnosed. So it has a higher death rate than almost any other cancer that's going, especially of the ones that afflict a lot of people. And so the number of diagnosed cases every year is almost equal to the number of fatalities. And so if you look at the survival rate, and actually this is a little bit more now, it's a little bit higher than 8%, but if you look at 5 years, you know, only 8% survival for those individuals. So, and the median age of survival, so the average length of time that somebody is going to survive, is less than a year. So it's very common to have someone come into the clinic, be diagnosed with pancreatic cancer, and within a year, they succumb to the disease. And so this is a devastating diagnosis that you can get. Big thing for us is that only about 5 to 10% of the patients who have pancreatic cancer are really due to some kind of genetic basis. And it's twofold, and this is a problem. One is that it's that you know, if what are the genetic basis of it? Well, we don't know all of them. We know a few, and I'll talk about a couple. But even in those cases, just because you have that mutation doesn't necessarily mean that you're going to succumb to pancreatic cancer. So it tells us that the genetics behind it plays a role, but not as significant as in some other types of cancer. And so there's a really strong relationship between the environment, and especially environmental stress. And as an example, another disease that afflicts the pancreas is chronic pancreatitis. And this is just a chronic disease of fibrosis. And what you see over time is that about 5% of those individuals, which is a fairly high rate, they develop pancreatic cancer. And so what are the challenges? I've obviously pointed at a few, but what are the ones that are specific? So this is a diagram or an image of what the pancreas normally looks like. And the big thing for us is that, you know, when we talk about the pancreas, and every time I talk to somebody about I work on the pancreas, they always ask about this thing right here, which is an islet of Langerhans. And this goes back to your histology. And this makes up only about 2% of the pancreas. The rest of this is pancreatic acinar cells. So this is what normally the pancreas looks like. And these acinar cells are basically producing all the digestive enzymes that allow you to digest your food. Well, if you look at it, there's these kind of precancerous lesions that develop. And actually, this is in a pancreatitis model. And you can see that you get these things that looked almost like ducts. See, they almost look like what's carrying the enzymes to the intestine, but they're not really. They're seeing this is kind of like a lobule right here. This is another lobule. And you see there's some damage that goes on. But then what happens is this is now pancreatic cancer. And the cancer really is this area here. So you can see that the cancer cells make only a small part of the tumor. All of these other things in here are fibrosis, like there's cancer-associated fibroblasts, there's inflammatory cells, there's neurons in there, but this is the majority of this is non-cancer. So when you think about a normal tumor, you think that, oh, this tumor just outgrows and you get this solid mass, but not so in pancreatic cancer. So that's the first challenge that we look at. The second thing is that you can get rid of almost all the pancreas. So this is an example of a model that we have, and this is a mouse, but I'll talk a little bit more about that. And this is histology. And if you remember, all of the acinar tissue made up most of the tissue. Here, it's only making up a very small portion. All of this other part of the pancreas that you see around here, and this is higher magnification. So this is normal and this is abnormal. These mice that we have look absolutely fine. They don't lose weight. They don't show any signs of any kind of problems. And yet you can see the majority of their pancreas is gone. And this can also translate to metastasis. So here's a normal liver on the left-hand side. And these are mice that are normal. And you can see these little blotches in here. Those are metastases. So pancreatic cancer not only takes over the pancreas, but it also metastasizes before you even know that you might have the disease. And so that proportion of individuals who are successful in recovering from pancreatic cancer are often detected early. And in some cases, they're detected because of a mistake, because they're being looked at for some other reason and they realize, oh, you have a tumor. Third challenge that we have is that if you remember back, I just told you about how the tumor, those cells are surrounded by other things. Well, they're surrounded by stroma and inflammatory cells that affect the chemosensitivity. And they also affect the tumor growth. And it's not just a simple, as you get more stroma, it becomes worse. There's a sweet spot almost. So here is an example. On the left-hand side, and what you see in red is what's called, this is collagen, and this is picoserous red staining. So anything you see in red here is fibrosis. Now, normally in a healthy tissue, you wouldn't see any of this. But you can see this has got some, and then as you go further to the right, there's a significant more fibrosis. Now, the problem with this is that, this fibrosis will prevent entry of, it could affect the entry of chemotherapeutic agents. It may also affect metastasis. So this might not be that invasive where this is, but this is something that we have to think about when we think about how you treat pancreatic cancer. And then lastly, pancreatic cancer is really heterogeneous. So there's two examples of this. So here's one example. And these, again, on the left-hand side is the histology of pancreatic cancer. And you can see right easily that there's a significant difference in this type of pancreatic cancer versus this. They're both adenocarcinoma, but this is called classical and this is called basal-like. This is much more aggressive than this, but this is not necessarily receptive to therapy. And there are different markers. So for instance, in this is immunohistochemistry. So we're looking for a protein, and we see that it's expressed high levels of GATA6. And this expresses high levels of this KARAT5. So we can say this is a classic-like tumor. This is a basal-like tumor. But knowing that will help us indicate what type of therapy they might get. But it gets even more complicated than that, because actually you have within the tumor a lot of heterogeneity as well. So this is a study that was carried out by a group in CERM at Marseille that I worked with. These are supposed to be all classic-like tumors. So these are actually cells that are taken out of the tumor. They're grown in three-dimensional matrices. And they look at them, and each one of these represents a different patient. They've all been diagnosed as classic or classical. They all have the expression of specific markers, and yet they all have these different types of morphologies that you see. And knowing this is going to affect how they may respond to therapy. So it may be that this one patient here, this patient 3, has a much more problematic tumor because there's a lot more cell heterogeneity than maybe patient 2, which shows a very simple type of morphology. And then below is the expression of a marker of a pancreatic cancer. You can see that here's the cancer right here and here. You can see that not all those cells express that marker. So if you're saying that this is a tumor and you're trying to target a certain pathway, if you target a pathway that's only active in half the cells, the other half of the cells are going to be able to basically escape that treatment. So this is things that we also need to address. Okay, so basically we need to develop early detection methods, because the earlier we detect it, just like any other cancer, the better off we'll be. We have to identify patient-specific therapies, and not only that they might be patient-specific, but when to use them, right? It's not just a matter of knowing in one snapshot, but maybe over time how that tumor might progress. And then we have to remove barriers to chemosensitivity. So this is some of the things that my lab addresses. So we use a different thing. So again, before I get into the actual modeling, I want to draw one more parallel. So I say at the end, there's this strong relationship between environmental stress and pancreatic cancer. And so we actually are trying to understand that process, because that happens in a lot of different ways. It's not just the initiation of the cancer, but you think about it, you have tumor cells that are interacting with other cell types. They're interacting with therapy, they're interacting with other environmental agents, and they are trying to survive. So they are trying to change. So we want to understand how they do that. And so what is the thing that it links this? And this is where we get to the idea of epigenetics. And I teach a course, I don't know, I think some of you might be in the 3rd year course I teach in cell physiology. And so if you've heard me talk about epigenetics, I hope it wasn't too traumatic an experience for you, but I'm going to hear a little bit more about it again. And so the idea behind this is you get changes in gene expression that have nothing to do with mutations in the DNA. They're essentially changes to how the chromatin is wrapped up, or how the genes are organized. And so you can change the expression by changing that organization. And so there's 3 main types of ways to do this. The first thing is you can modify the DNA base pairs. And you can talk about these as that being basically you take a base and you modify it by methylating it. So this is a direct change in the structure of the DNA, and it usually represses the genes where you see methylation occurs. The thing that, and this is something that we spend most of our time with, this is the thing, is how is that DNA packaged? It's normally packaged around these nucleosomes. So these bubbles that you see here are proteins, and DNA is wrapped around them, and you can modify these, and in doing so, you basically can either open up the DNA or you can close the DNA. And then finally, you can produce a whole bunch of microRNAs, or actually we can expand this to RNAs that don't make protein. RNAs that do something other than that, where they may actually affect, again, the way the DNA is wrapped. So we try to understand some of these things, okay? And so if you think about this in the simplest way, this is your histones that the DNA is wrapped around, and you have these different genes that are next to it. You can see that if you have these things and they're open, they're spread out, that these things can be transcribed. RNA can be made on those genes, right? But what happens is epigenetic reprogramming will switch these things. And this happens normally in development, but any environmental exposure can do the same thing. And so what you might see is you might have this gene A where it's exposed to something that causes it to become much more condensed. And that turns off that gene. And again, that could be something as simple as a dietary change. Could be good, could be bad. We have to figure out what's going on. And in the same way, you could have genes that basically become more open and they become active. So in the context of cancer, you would say potentially what's happening is this might be a tumor suppressor gene. And it's very common that you get a tumor suppressor gene that becomes closed, that becomes condensed, and therefore you don't see expression and the potential for oncogenesis occurs. And these genes might be pro-oncogenic. So they would be, tumor initiating or tumor promoting genes. So we try to understand how those fit into it. And we use a couple of different models, and I'll talk about those now. But last night, and I've already made this point, so just kind of show it. These are all the different things that can affect the epigenome. It can be really anything. And so we try to understand, really how therapeutic drugs, how diet or, exposure, it could be drugs, could be cigarettes, could be stuff like that, either reprograms it to increase susceptibility or reprograms the genome to affect sensitivity to chemotherapy. So those are kind of the things we do. So this is kind of, to put it in context of where my lab is that this is a schematic just showing that here's your acinar cells. So that's what makes up the majority of the pancreas. Those are those, circular things that I showed at the beginning. Here's the duct that goes in, and it undergoes this transformation to become like, this is basically what we call ADM, or acinar ductal metaplasia. This is the very initiating event of pancreatic cancer. It then goes on, forms these intraneoplasic lesions where they're going to actually become cancer, and all of these cells around it are either, cancer-associated fibroblasts or tumor-associated inflammatory cells, and the red is fibrosis. And so the things that we ask in the lab, how does chronic stress affect this? How does, if you have chronic stress on these cells to make this more susceptible to this later progression, what are the actual changes that occur there? What are the things that are affecting that? And we look at a pathway called the unfolded protein response, which is essentially a stress response pathway that's there to allow the cell to survive, but in doing so, may enhance this progression. We also look at specific genes that regulate whether genes are off or on, so these transcription factors, and how that drives this process. We try to understand the interplay between the cells that are outside the tumor and the tumor themselves. There's a lot of really good evidence that's being done by a number of labs around the world that shows that macrophages in this area will actually suppress an immune response to try and get rid of the cells. So people are actually trying to get rid of the macrophages so that they can have lymphocytes come in and actually target and destroy the tumor cells. And then finally, we're trying to understand how chemo resistance is regulated, or how that leads to epigenetic reprogramming. So next two sections, and this is basically the idea of what models we use. So the first model that we use extensively in my lab are mouse models. And so I always have this schematic up, and it just kind of talks about how you can make a genetic model of pancreatic cancer. And I know this is, again, maybe something where you might be saying, I didn't want to get a genetics lecture, but here it is anyway. So here is a gene that really promotes pancreatic cancer. This KRAS, it's been known to be mutated in like 95% of pancreatic cancers, and it is considered a primary driver. And the idea is, what we do is we don't want that to be expressed right away, so we put it in the context. So this is the gene. We put it in the context where we have these what are called LOXP sites that sit on either side of a stop codon. So the idea is that this gene is there, but because this stop codon's there, no KRAS or oncogenic KRAS is made. But then we made it with a mouse that now expresses something from a gene that's expressed in the pancreas. So in this case, this gene is always expressed in the pancreas and only in acinar cells, but it produces this thing called Cre recombinase that's inducible. Now we mate these two mice together, we get a mouse that has both of these genetic events. And then if we give them something called tamoxifen, what it does is it allows this particular protein to go to the nucleus, and it actually cleaves out the stop codon and turns on KRAS. So now we basically saying we're going to turn on KRAS and promote oncogenesis at this point in time. So again, here's your acinar cells. Here's the normal way it looks like. And we say, okay, we're going to now turn on KRAS, and we get these pre-neoplastic lesions. And in the majority of cases, that's as far as this goes. So these really aren't, this isn't really cancer. So there's something else that's going on that's driving this process. And so this is where we say, okay, well, we're going to try and see if we can put in other genetic manipulations, expose these animals to other factors, do whatever we can, see what promotes cancer, what accentuates cancer, and see what prevents cancer. And so just as an example, if we then go and provide an injury that's similar to pancreatitis, now we have significantly more lesions, and this is the beginning of pancreatic cancer. So this is how we use this model. And we tested a few ways. So this is an example here. We published this a few years ago, and this was a graduate student, a master's student in my lab that did this project, and she got a publication on it in a very good journal. And what she noticed, using that system that I just talked about, that if she combined the activation of KRAS with a mutation in this one epigenetic gene called ATRX, if you got rid of the ATRX and activated KRAS, that you ended up getting cancer forming, but only in a subset of mice. And it turns out that that subset of mice was female mice versus male mice. So really, you can see here again, this is a male mouse. These are the exact same process. They both, have activated KRAS, so oncogenic KRAS. They both have a loss of ATRX, but in this case, no difference from the wild type. This is wild type tissue, but in the female, significant progression of pancreatic cancer. And we actually verified that if you look at patient samples, if you ever see an ATRX deletion or a mutation in a pancreatic cancer sample, it's almost always, or in our case, we always saw it in a female patient. So it's kind of interesting to see that. So here we showed that this deletion basically enhanced sensitivity, but in a gender-specific fashion. We also take this another model. We're like, okay, now we're trying to understand the therapy. So, and trying to understand not so much therapy, but like, can we identify the expression of some of these factors using imaging? And so here's an example. So this EZH2, and it doesn't really matter what this is, but it's a protein that is involved in epigenetic reprogramming. So it goes on and it actually silences genes, and it's highly expressed in some forms of pancreatic cancer. And so the theory is that maybe we can use this as a therapy. But the thing that you don't want to do is you don't want to give somebody a therapy just because you think it might work. You want to have some evidence that it might work. And so the idea for this is to actually try and develop an imaging molecule, and it's based on an inhibitor for this protein. So this is the inhibitor for EZH2. And we modify it slightly. What we do is we add a fluorine molecule, you can see at the one point here. And this allows us to do PET imaging. And then we can say, are we seeing accumulation of EZH2 using the PET imaging? So here is a mouse. This is really no, not the Grace imaging, because all of this here is just background. But this is where we put a tumor in. And so you can see that there's some accumulation in here. And actually, if we then compete away this fluorinated molecule with the parental molecule, so it doesn't have any fluorine, then we can actually see that we lose that imaging. So this is obviously a work in progress, but this tells us that we might be able to target and identify whether EZH2 is expressed in tumors before we might want to go on and try and treat them for that with that type of therapy. So we can have new ways using mouse models. We can use, develop these new ways for detecting gene expression. Oh, and by the way, this is a really nice collaboration that's ongoing with somebody in chemistry. Again, if somebody told me 10 years ago I'd be working with a chemist, I would have laughed, because chemistry is definitely not something that I know nor really enjoy, because all of this stuff up here to me is just, squares and squiggles and stuff like that. So then we can do other things. So again, here is this, these two animal models, and we have this model that we say, okay, this is now activating KRAS, and we get this progression of pancreatic cancer. And if we injure the animal and adding pancreatitis, we get even further. And here's a situation. Now we've modified one of the gene. We've gotten rid of a gene, and in two ways. It's reduced the amount of progression, and it's reduced really the maturation of these lesions that are going to form cancer. But the thing is, we don't know if it's because of something that's going on in the tumor or something going around in the cells around it. And so we do something called CyTOF, but really what it is it's basically identifying cell populations based on the surface markers that they have. And so this is kind of the end point of what we do. You basically dissociate the whole tissue, you get single cells, and then you see how they express surface cell markers, and then you could group them. So all of the cells in this group here are the same, all the cells in this are the same. And knowing what cell surface markers you're looking at, you can tell what types of cells they are. So you can see that the majority of these are different inflammatory cells. There's also epithelial cells, which are essentially the tumor and these fibroblasts. So those are the epithelial cells right in the middle. But what you can see is if you compare this one population right here in pink, there's a lot more in this animal than there is in this animal. These are macrophages. And I said already that macrophages essentially block progression of the tumors. And so that's maybe what's going on here, is that this effect is not necessarily in the acinar cells themselves or in the tumor cells themselves, but it's something around them that's going on. And the other thing we can do is each one of these dots that's in these things indicates a single cell. Well, now we have the ability to go and take that single cell and sequence it and understand how the expression pattern might change for each different cell type, or if there are different cell types within this population. So we can basically identify how these different cell types can be affected to the genetic deletion or treatment, etc. Okay, so finally talking about how we now have a project that's ongoing with a number of clinicians, and these are surgeons and GI and endoscopists, where we can take human-derived tissue and actually still model disease, but do so in a patient-specific way. So the whole idea is that you have an individual patient who comes in, and that person has cancer, and everybody who has pancreatic cancer or any type of cancer, they don't have the same exact disease. It's always something that's, there's going to be differences based on their genetic background, based on the exposures that they had. So they may not all respond to the same therapy, or they may not respond to any therapy that we know yet. And so what we can do here is we can take this biopsy and we can take it either from the tumor itself at the time of surgery, or we can take it from endoscopy. So we actually, before there is a diagnosis, we can have the GI physicians go sample from the suspected tumor, give us this material, and then we grow it in culture and expand them. And then we can actually look at their morphology, look at the phenotype, their gene expression, and then even set it up where we can potentially do some screening and may be able to inform the physicians, hey, this individual is probably going to respond to this therapy versus this therapy. So here's some examples. So this is just, normal tissue. You put it in this three-dimensional culture and you see, there's maybe a little, like that might be a little bit of a cyst forming, not much. But if they have some tumors forming, you can see that they grow fairly better. There's this going on. These are from tumor tissue. But then if we get an aggressive tumor tissue, this is the things that we see. And this is the same magnification. So this isn't like I made this magnify. This is just that much larger. And so we can see that just within a couple of days of doing the culture, you can see this difference. And the other thing that we can see is we can really get to the heterogeneity. So here is what we call an organoid culture from endoscopy. We have all of this tissue here. It's all in 3D. And you can see, even just looking at it, there's a lot of different types of things that you're seeing here. So this is kind of a group of cells that's form a cyst. But then you see over here, there's a different morphology. There's some that are just small. But the thing is, if you put them on plastic, they do something completely different. They show this kind of significant different morphology. And again, this is stuff that we can now take and test. And so the last thing I wanted to kind of show you is how they respond. So here's a project that's been ongoing in my lab where we look at that unfolded protein response. I talked about this before. It's a stress response that protects the cells. And what we do is basically we say, okay, so this middle line here, and I apologize for the color because the color is mixed up, but the middle line here is how those cells respond to normal therapy. So if we take something called gemcitabine, which is a standard therapy for pancreatic patients, this is the responsiveness of those cells. So as you see it decreases, that means the cell number, they're more susceptible to undergoing apoptosis. If we treat with a molecule that inhibits the unfolded protein response on its own, it does nothing here. And even here, it has it does tail off eventually, but you'd never use this at this type of concentration. But if you now combine those therapies, you're seeing a dramatic shift, especially and it's done in a patient-specific way. In this particular case, it's a very dramatic shift. So these are much more sensitive to the combined therapy. And even in this, there's a shift in sensitivity. So this is kind of the beginning of trying to understand combinatorial approaches to how we would be able to treat patients in a patient-specific way. Okay. So just to kind of finish up and to kind of talk about the ongoing project, I talked about in general what we do, but we have a number of projects in the lab, and the people here are, you can see, are associated to it that are currently in the lab. So we do something in calcium and pancreatic disease, and I have a postdoctoral fellow and an MSc candidate and even undergrad student that's working on this. We do this transcriptional targeting, and I don't know if they're here yet. I'm going to actually see if any of them have shown up. I know that one person here is, I think Fatima is in the room. Yeah, so Fatima's here. If you have any questions specifically about graduate studies to her. But they do this transcriptional targeting in pancreatic cancer, looking at the unfolded protein response. And then we have a nice project in epigenetics of pancreatic cancer. I talked about some of it, but we have, same most models looking at the sensitivity of that. And then I talked about the imaging epigenetics. It's a collaboration with Len Lewis. So with that, I'll stop, and I will be happy. And I know that you can probably have questions for either Dr. Barbeau or myself, or even, as I said, you can ask Fatima Musavi, who's on the line, and anything about why she got into graduate work or things like that.
Well, thank you so much to both of you. That was really, really cool. I know we get to hear from you in class, but it's always wonderful to hear kind of, what you do in your day-to-day life. But yeah, of course, we'll be opening it up to questions. So, feel free to unmute or leave it in the chat, whatever you feel most comfortable with. Yeah, I guess take it away.
I have a question directed more so, I think, at Dr. Barbeau about the clinical anatomy master's program. It's actually one of the programs I'm looking into. So I was wondering if you could talk a little bit more about that, and maybe in particular, like how placements, so to say, would work, or how you get assigned to a project when you enter a master's like that.
So that program is a little bit different than our research stream. That's sort of the way we call them the clinical anatomy students and the sort of classical research master's program. And those, for the research projects in the clinical anatomy program, the researchers submit their project to the course coordinator. Basically, the research project is actually a course. And so we submit what we want the student to do for this project. And then the students have a chance to see all of them. And then they approach once they're accepted into the program, they approach, and basically it's sort of interviews. So we do a match. So the student interviews at least 3 researchers, and then they rank their preference of project, and then it sort of gets matched up like that. So the project is pretty well laid out. You can take it a little bit in the direction that really sort of interests you somewhat, but we have a really tight time frame because that program is 20 months. The first 8 months is basically pretty intensive coursework. And then, so basically from the end of April until for 1 year is when you have to get this project done, which is a pretty tight time frame for a research project. The research stream, those students only have a couple of courses to take, and then they sort of have 2 years to complete their research project. So does that answer? Did you have specific questions about?
No, I think that did a really good job of answering that. Thank you.
Actually,
I have
sorry, Jasmine.
Right. Sorry, Blaine. I just have a quick question for Dr. Pin. So like in this year, like you've kind of gotten more familiar with like experimental procedures and like different laboratory techniques. So I was just kind of wondering which specific techniques do you use most often in your day-to-day research?
So that's a hard question to answer because it really depends on the project. I mean, one thing that the people in my lab do a lot of is they do a lot of histology with regard to just normal staining, and then immunohistochemistry and immunofluorescence. But it's usually coupled with other things, right? So the methodology might be working with mice. You're doing an experiment where you're you're treating mice with something. You might be doing, if you have a tissue culture project, you're gonna be in culture pretty much every day. But we also extend to other things. So we have people in my lab who do bioinformatic analysis. So they're looking at large data sets. So their day-to-day is a little different than the people who are, basically doing a calcium imaging project where they're looking at, doing microscopy. So it really depends on the lab that you're working in, but I would say that all of the stuff that I showed you, those are things that were done by graduate students. So yeah, a lot of cell culture, a lot of immunofluorescence, as I said, bioinformatics, things like that.
Okay, thank you so much. Yeah, that was really helpful.
I see there's a question from Connor for me. They mentioned there's a goal for personalized medicine to become a more common practice. Do you have insight on the timeline for this to occur? I would like it to occur yesterday. It really depends on the disease. So I know that this already is in practice for some cancers. It really depends on how well characterized the therapies are. I will tell you for pancreatic cancer that it's still a little ways away. Right now, what happens is a patient comes in and they basically say, okay, is this patient in relatively good health or relatively poor health? If they're in relatively poor health, they put them on a therapy that's generally a single molecule. It's like, gemcitabine might be it. There's also paclitaxel, which are just, brand names for different things. And the reason is they don't think they'll be able to handle the more aggressive therapies that might be better. So if they're in good health, they give them something called FOLFIRINOX, which is actually a mixture of 4 drugs. But if you give that to somebody who's not in great health, they're probably not going to survive it. So that's how they distinguish what type of therapy to give somebody right now. And then if they don't respond, they say, okay, let's try something else. So they don't really do a lot of pre-screening for pancreatic cancer, and that's where we want to get to. For other ones, I think you might see this in breast cancer. They actually do some genetic analysis because they can say, based on a patient and the types of genetic mutations that occur, that they'll respond to one type of therapy versus another. So it's already ongoing for some diseases, but for other ones, we're still a little ways away.
I kind of have a question for both Dr. Barbeau and Dr. Penn. In your academic history, would you have preferred either for Dr. Barbeau to go straight into your PhD or for Dr. Penn to have done a master's before your PhD?
I'll let you answer that first, Michelle.
No, it just kind of worked out. I'm happy the way things worked out. I wasn't ready to do a PhD when I finished my master's. It's a fairly big commitment, and I guess I maybe didn't have enough confidence that I thought I could keep thinking up new experiments, and so I was happy to kind of get the teaching experience under my belt, and then a PhD is it's a pretty powerful degree to have. It can take you into academia, but it can also take you other places as well, which I don't think we do as good a job promoting all the other things that you can do with a PhD. But looking back, I really wouldn't have changed it. It did allow me I had little kids and my husband was pretty busy. So I was sort of taking care of my kids. So it kind of allowed me to do everything. So I didn't have it all at once, but eventually I kind of had stuff. So I'm pretty okay with the way things have turned out.
I think it's funny because when we were graduate students together, I would have said that Dr. Barbeau was much more on top of things than most graduate students and probably better able to handle the life of an academic more than most of us. So I'm going to disagree with your assessment of yourself. So anyway, for me, so this is case dependent, and I think you have to decide what you're happy about. I mean, for me, I didn't plan on doing a PhD when I started my master's, but I really enjoyed the project I was working on. And so that's why I tell my students that if you're thinking about switching to a PhD, you have to really like the project you're on. If you don't like the project you're on, then finish it and stop and do something else. And also you have to get used to the type of things there are in research. If you're okay with not always having answers right away, if you're okay with, working, some long hours, it doesn't have to always be long hours. It doesn't have to be always like a 70-hour work week, but there are going to be some times, and that you get excited about that, then those are things that tell you should be able to do it. Having said that, I know a number of really good faculty members who have very good life-work balance. And so it's possible to do that. You just have to be a very organized individual. So all of these things make it very case dependent. So for me, it was definitely the choice to do it, to not worry about going and doing, taking time off between my master's and my PhD. I fast tracked for, a lot of the stuff, and for me it worked, but I don't think it works for everybody.
Thank you. I actually have another question for Dr. Barbeau. If you could go back and like kind of talk to your younger self about like giving yourself more confidence, I guess, to do a PhD at a quicker rate, what would you tell yourself?
I know what I would tell you.
Like I ask mostly because I feel kind of the same way, that like I'm probably not capable, but I know I probably am, but it's hard to kind of like tell yourself you are.
Yeah. You don't do a PhD in like 1 year. So you sort of plug away at it and you keep moving forward. So you have a plan, you have a good team, I would say. So your advisors and your supervisor is probably the most important thing. So if you have people around you who are really supportive, like this isn't what I would go back and tell myself, but this is what I would look for if I was advising people on what to look for. I would also advise you to start with a master's for sure. I think there are opportunities for you to go into a direct entry, but if you have not had a lot of experience with research, a master's is a fantastic way to find out about it, to see if it's for you. And a lot of programs do let you switch.
Mm-hmm. So I'm going to answer that too, because I've trained, I think more than 20 graduate students now. About a quarter of them have been PhD students. And I think that the majority of people can do a PhD. It's really about perseverance, and it's really about support. So again, if you have a really supportive lab and supportive supervisor, it becomes that much easier. But it's really about just being patient, and some days are going to be good and some days aren't going to be good. And yeah, I think everybody should start in a master's if you're going to start a graduate degree. And if you want to switch within 16 months, every program that's at Western has that ability to switch over. And generally you don't switch over if things are not going well or you don't like it. You only switch if things are going well.
Thank you both very much.
In the chat. Oh, you're welcome. You have another question in the chat. It's for you, Michelle. I think you're on mute. You're on mute.
I just read the question. So is previous research required for the clinical anatomy? No. For the other stream, not technically, but a lot of students do have, a lot of students kind of do a 4th year project with someone, and then a lot of times that turns into a master's. But it's not necessary, no. You do have to want to do research though, so you're not avoiding research. So I think there's a little bit of I don't like to do research sort of feeling. There's still a significant research component in it, and research is a lot of fun and you shouldn't be afraid of it.
I guess since we're talking about grad programs, I figured Dr. Penn's grad student, if you'd like to introduce yourself and maybe talk about like whether or not you like your experience at the moment, or kind of a bit about your program at the moment.
Hello, hi. Thank you. Yeah, I'm Fatima, and I'm a 1st year master's student at Dr. Penn's lab. And I'm very much enjoying my experience in the lab. It's funny because when I was in undergrad, I was pretty certain that research was not my thing. I was always interested in cancer. Like, I thought maybe I'll become a physician and I'll work in that field. But I had a chance to go to the UK and I did a cancer biology course. And that was really when I found out that there are really more questions. Like, there has been great progress in the past few decades in the field of cancer, but there are more questions than there are answers. And I wanted to be part of a group that tries to answer some of those questions. So that's why I joined Dr. Pin's lab, and it's been great so far.
Fatima, why did you think research wasn't for you before you entered the program?
I think maybe I didn't have as many positive experiences being part of research labs in undergrad. I mean, and I'm not blaming anyone. I think part of it was just me being very anxious in labs, being around lab equipment and doing things and being scared of messing things up and ruining other people's projects and things like that. I think that was, I mean, being an undergrad, I felt like I was kind of a guest in every lab I went. It was never my project that I was doing. So that was a fear that I always had, but it's certainly, like, it's very different when you become a graduate student.
I think that, and as you can tell, Fatima's in the lab because she has a mask on, so she needs to have that on when we're in the labs. But I think it really comes for people who are undergrads. You don't appreciate what it's like to be a grad student because, first of all, you get paid. You have that. It's a year-round thing, so it's not like you have a summer break, but you still get some time off. I think the majority of labs have it now where if when we get out of the pandemic, and this is before, is that you do a little bit of traveling to different meetings if you're presenting. And that's kind of some of the rewards. You talk about the things, you actually can go to meetings and actually interact with some people and actually listen to some of the really best researchers in the world. I remember when I was an undergrad and I went to Boston for a meeting in cell biology. And I got to listen to, a Nobel laureate talk about their research and talk about not just about the research that made them a Nobel laureate, but the work that they were doing that wasn't published yet. And I thought that was so cool. And then the other thing that's really cool, and I tell this to my students if they ask me why I got into research, and I say, for a moment in time when you do something and you get a result, you're the only person in the world that knows it. That's a pretty cool feeling. It's a pretty cool feeling. And then when you see that eventually published, then it's even better.
Wow, that's a really actually impactful quote. I never really thought of it that way. So we were just going to say thank you for all the great questions, and especially thank you to Dr. Barbeau and Dr. Penn for coming today. It was super insightful and very helpful to see like all the different avenues that you can take in research and things like that, and also with anatomy and cell bio. In the interest of time, if you have any more questions, please feel free to message us or the Western ACBC social media accounts, and we can connect you with either of the professors for further questions. We also recorded the session and are hoping to post it on our club website or on YouTube, so keep an eye out for that. And until then, thank you for coming, and we hope you have a great rest of your week.
Thank you again.
Thanks for